One of the projects that we've done was basically doing what I described, and this is a project called Harmonizome. It's a website that you can find in this URL. And the idea is that we went to all of those resources and we processed the data, wrote some Python scripts to process the data, and it has now 72, almost 72 million associations between terms and genes. So sort of like exactly what I described, and it's presented nicely in a search engine where you have gene pages and term pages, but now you can find a lot of information that was embedded into those data sets that was not really readily available for people to see. So you can see which genes, when you knock them down, are affecting the viability of some cell lines. Or one thing that is added to this that makes it a little bit more complicated, also those genes have weights. So it's not just like the way I describe it. You can imagine that every gene has a weight of 1. So this, in sort of like mathematical terms, those are sets, because you have a set of elements that belong to some label, and then they all have the same weight, and the order of them is not important. You can shuffle them and it will still be the same object. So this is defined as a set. If you make the membership, so this one represents the level of membership. So in a regular set, the membership is always 1, but in a fuzzy set, you can have a graded membership that is between 0 and 1. So every gene has some number that defines its membership to the set. Okay? So here it gives you, in some cases, when we know, let's say if a gene is really killing the cells by a lot, we can grade that compared to a gene that, let's say, does like partial killing of the cell. So, or a gene is highly differentially expressed, or so we can add this quantitative level to those sets. Okay. The same approach is also used for this tool called Enricher. This is like our most famous tool that is used by a lot of people at Mount Sinai for their research and other people in the world. And then it does enrichment analysis against a bunch of libraries. So we have 143 libraries, gene set libraries, that are stored in this type of format. Beyond serving data to the community, providing this resource to the community, it opens the opportunity to do a machine learning or imputation of knowledge. And in this particular case, we took the data that we processed from all of those resources, and we took some transcriptomics data. Some transcriptomics data is from tissues, like basal expression across tissues. Some transcriptomic data is from perturbations, so like looking at whether a gene was up or down under different conditions. And then here it's like some proteomics data and some pathway data. So the pathway data is binary, whether the gene is in a pathway or the protein is in a pathway. But these guys have they're like continuous variables. So you can concatenate all those types of data sets where the genes are the rows and then those attributes are the columns. And then we want to predict whether we're going to see a specific mouse phenotype if we knock out a gene that hasn't been knocked out in a mouse yet. So one of my favorite mouse knockout phenotypes is increasing the mouse's age. So some genes, if you knock them out in a mouse, the mice may live much longer. Potentially there are human genes that can make us, if we knock them out in human, potentially will make us live longer. So not all genes were knocked out in mice yet. So let's ask the interesting question, can we predict which additional genes, if we knock them out in a mouse, will extend the mouse lifespan? So we have a bunch of positive examples. We have a lot of negative examples of genes that were knocked out in a mouse. The mouse lived the same lifespan or died early. And then we have all of those genes that were not tested. So we can build a function that takes all this data and produce a score or prediction of how likely it is that each of those genes not tested will be our candidate. And because those experiments are very expensive, it might be useful for us to know which ones we should prioritize. And this is just one example of how you can take all of this process data that I showed you and apply this machine learning to make predictions and prioritize your next set of experiments. Now there is very easy to use libraries that you can learn how to use, like scikit-learn, TensorFlow, and also this deep learning, which is artificial neural networks, is widely applied, not necessarily to these type of problems, but it's now sort of like the state of the art. And you hear it on the news, how it solves a bunch of complex problems, and it's sort of like a resurrection of artificial intelligence and machine learning.